This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. We are in the 6th chapter, molecular basis of inheritance. So now we are going through the main portions of this chapter, important topics of this chapter. You can expect so many questions for NEET and for your board exam from this chapter. It's very important and it's dealing with very important topic, basic concepts of biology. So here in this chapter, molecular basis of inheritance, we have learned about the DNA, then RNA, replication of this, then transcription, genetic code, translation. All these we have learned now. How replication is happening, then transcription, how it is taking place, then what is the genetic code, then how the genetic code is making a protein, or how a protein is getting formed, or the formation of a protein. That also we have learned, right? So in today's class, we are going to learn about regulation of gene expression. Okay, regulation of gene expression. So from that title itself, it's clear. Regulation of gene expression. Gene expression is getting regulated, or it is getting controlled, or the expression of that gene that we are going to regulate. So that's what we are going to talk about. So actually, what is the gene expression, or what do you mean by gene expression? Expression of a gene. And you know that, now you know that how a gene is getting expressed, right? In the previous classes, we have discussed about the expression of a gene. So how a gene is getting expressed in one organism. What is a gene expression? What do you mean by a gene expression in an organism? So gene expression means information from a gene. You know that all informations are present in a gene. So this information is used in the synthesis of a functional gene product. So a gene that can't directly act, or that cannot work directly in an organism. So in that place, it is creating, or it is forming a functional product. It is forming a product which can function, which can act on the body of an organism. And it can create some particular changes, or however that gene is, or the wish, how it is wishing to act in the same form. That's functional product, that product which is functional, that can act on the body of an organism or in a cell or whatever it may be. It can function in a particular way in that organism. So what do you mean by gene expression? That means information from a gene is used in the synthesis of a functional gene product. So for the synthesis of a functional gene product, the information from a particular gene is used. That is called as gene expression. So how that informations are getting expressed? That information which are present in the gene, it is getting expressed in the form of a product which is functional. So in the form of a functional product, a gene expression is taking place. And this gene product which is formed, that product is functional, and that will help to produce an end product, or a protein, or a non-coding RNA. Okay, that may form a protein, or it may form a functional product that can act as it, that may form an end product. Okay, or a non-coding RNA that may form a protein, or sometimes it may not. Okay, so finally that forms. It forms or it produces some effects in that phenotype. Okay, so what happens? it produces some kind of effects in that phenotype. Phenotype means the visible character of an organism. Phenotype and genotype we have learned. So what is the phenotype? That expressed form or that visible form. See, a plant is a tall one, an organism it is, or the hair color. So like that, hair color is dark, it's so black, or sometimes it is white. So depending on that character, we can say that is the final expression, right? So a protein that codes for sometimes brown hair, sometimes it codes for that gene, particular gene codes for dark or black hair. Okay, so such things, the final expression, how it is getting appeared, that is a final expression. Okay, so the product which is functional, that will help to produce an end product. And this end product or a protein, sometimes that may be a protein or sometimes it may not code, or not, sometimes it may be a non-coding RNA. Okay. So finally, it forms or that it produces that gene or that particular gene produces some effects in the phenotype. That product or the gene produces some product that may be a functional, that is a functional product, and that end product that may be a protein or maybe a non-coding RNA or whatever may be, that can create some kind of changes in that particular phenotype. So that is the final or ultimate effect of that gene. Okay. So a gene is having some kind of expression. That's what we are talking about. So how this expression of a gene is getting regulated or how it is getting controlled. Now we are talking about, just now I said about the expression of a gene. In some way it is getting expressed. Okay. So how this expression is getting controlled. So regulation of gene expression, that includes wide range of mechanisms. Okay. It is having wide range of mechanism. Regulation of gene expression means that is not a simple thing. It's that is having several mechanisms, like wide range of mechanisms. So many things are included in that. So how it is working? How this regulation of gene expression works? These mechanisms are used by the cell. Okay. So this regulation of gene expression, all these mechanisms, just now I said there is wide range of mechanisms. Several mechanisms are included in this or involved in this. So all these we can use, all these are used by that particular cell. Okay. It is used by that cell to increase or decrease the production of that particular gene production. Okay. Or that particular protein. How that a character is getting expressed or how that final expression is, it's in the form of a protein or a final product. And that final product makes changes in the phenotype, right? That particular protein or that final functional product is making some kind of changes in the phenotype. So the regulation mechanism, regulation of this particular gene expression, that expression of that gene is getting regulated. So that may increase the production. Increase the production of that particular protein or that final functional product, or it may decrease. See, from a normal gene expression, if it is getting increased, that expression, gene expression will be different. If it is getting decreased, the gene expression will be different, right? If you don't want one character in a particular plant or in an animal, so in a normal gene expression, a protein is getting synthesized, and that protein is making a final phenotypic expression, and we don't want that particular character. So what we should do? We should reduce the synthesis of that particular protein so that that expression, that phenotypic expression won't be there, or it will get reduced, right? So like that, we can control the gene expression. So you know that what is a gene, right? So a gene means a part of DNA, and that part of DNA, that particular part that can code for a specific protein. Okay. So in DNA, all the informations of a cell, it is stored in that particular DNA, isn't it? So this information which is present in the nucleus, and this DNA is carrying all these instructions with the which is required for that particular protein synthesis, and that protein synthesis that is needed by the cell, right? So where that informations are present, it is present in the DNA, and all these instructions are needed for that protein synthesis, and this protein is required for that particular cell. Okay. So for each protein, there is a specific function in the organism in the cell of an organism, right? So each protein is having a particular function or a specific function in the body, right? So each gene will have full set of instruction, and these are in the form of genetic code. How that instructions will be present? It is in the form of genetic code. Gene means the genetic code. Okay. See, before explaining this regulation of gene expression, I'm just coming to that point. I'm just explaining gene and how it is working, how it is synthesizing a protein, and how we can control or how we can regulate the expression of that gene. Okay. So for each protein, there is a specific function, and each gene will have full set of instructions. And how that instructions will be present? These instructions will be present in the form of genetic code, and these genetic codes are transformed in the form of a protein during which step? During translation. Translation is the protein synthesizing step, right? So in that step, these genetic codes, these codes are read by the mRNA, and finally it codes for a protein during translation. So how this is happening? The genes first, they are transcribed into mRNA, then mRNA is getting converted. Here you can see different steps: transcription, translation, all this. First DNA replication, then transcription, then translation. Okay, so transcription means mRNA, then from that translation, this mRNA into protein. Okay, then after transcription also processes are there. Then after translation also some processes are there. Okay, that we will call as after transcription, post-transcriptional processing. Then this translation step will come. Then after translation, post-translational processing. Okay. So first, the genes first they are transcribed into mRNA. Then comes the transcription. Then this mRNA is getting converted into polypeptide chain. Then this polypeptide chain is forming the protein. Okay. mRNA first, gene to mRNA, then mRNA to polypeptide chain, and this polypeptide chain, it is forming the protein. So all the genetic codes which is expressed as physical traits, these are called as expressed genes. Okay, expressed genes which are expressed. So all the genetic codes which is expressed as a physical trait. So which and all genetic codes are expressed as a physical trait. With each the genetic codes, it will form protein. That protein will express as, or it will give a physical expression or a physical trait. These are called as expressed genes. Those genes are expressed as physical traits. Or otherwise, it is called as expression of a gene or a gene expression. So here, a gene or a genetic code, those genetic codes are managed in the protein synthesis. Okay, so the genetic codes are managed in the protein synthesis. So the gene which are having the information for an amino acid sequence formation, they are called as genes which are having amino acid sequence for a protein synthesis, or they are having information for an amino acid sequence formation. See, a particular some genes they are having, some genes they'll code for a protein, some won't code for a protein. We know that. Okay, no coding and non-coding. So that coding, the genes which are coding or which are having the information for an amino acid or an amino acid sequence, that is called as structural genes. So the coding genes, they are called as structural genes. The coding gene or the gene which are having information for a particular amino acid sequence, that is called as structural genes. So at various levels of a protein synthesis, we can control the expression level of a gene. Got it? So we can control the gene expression at different levels. Okay, so various levels of protein synthesis. There are several steps in the synthesis of a protein. From the starting stage onwards, and several steps are there. So in all these steps, we can control the expression level of a gene, isn't it? So the gene expression and regulation, it is not a single step process. I've already told there are several steps. So the gene expression and gene expression of and regulation of this gene expression, it is not a single step. And as I said, this is a very broad topic for 12th standard students. What and all points are required, only that much I'm going to explain now, or else you may get confused. Okay, so listen carefully, note down which and all points I'm explaining, note down all these things. So you know that gene expression, what do you mean by gene expression? Gene expression means the polypeptide chain formation. In that polypeptide chain formation is there, right? That amino acid sequence, it will form polypeptide chain. So this step was involved. So this can be regulated in several levels, right? If you want to regulate, or we can, if you want to control the expression of a gene, that amino acid sequence formation or that polypeptide sequence formation, that we can control or that we can regulate. In several steps, we can regulate this. So just for a simple example, just in our common life, we can say some example. See, if I don't like the discussion of politics, I don't like that topic, the political topic discussion. So if someone is discussing about that, I may get irritated, or I may blast, I may shout. I can say that don't discuss this topic. Okay, several ways are there. Either I can shout there, I can blast there. So how can I avoid this situation? Or if I'm getting angry, how can I avoid that kind of situations? Just like a common example you take. So in different ways I can do. In one way, I can avoid such discussions. See, before entering into such discussions, I can avoid this, right? So no chance to get angry. If there is no political discussion, which topic I don't like, there is no chance of getting angry, right? See, by chance I'm getting in between some people who are interested in such discussion, or they're interested to discuss about those topics. Then what I can do, because I don't like that topic, so what I can do, I can keep quiet, or I can walk away from that place. I can avoid that kind of situation. So like this, before that incident, I can avoid. Or up to the last moment, I have several chances to avoid that, right? So like that, in the same way, the expression, the gene expression, or before the synthesis phenotypic expression with the help of that particular protein, we can control at different levels. Okay, if that particular protein is forming, then only that is getting expressed, right? Or that phenotypic character will form. So that protein, if that level or that amount of that protein, particular protein synthesized, it is less means it will give another expression. If it is more means it will give another expression. Okay. So like that, for the regulation of expression of a gene, we can control at different level, we can control the regulation of gene expression. Or the gene expression we can regulate at different levels. So in the case of eukaryotes, here now we are going to talk about eukaryotes. In eukaryotes, all steps we can control. In which and all steps we can control this gene expression? Do you have any idea? So in eukaryotes, that regulation of gene expression can be done at transcriptional level. Here, transcriptional level. Transcriptional level means mRNA. In that level we can control. Then processing level. In the processing level we can control. Then while transport of mRNA from nucleus to the cytoplasm, that time we can control. This mRNA from nucleus, it should reach cytoplasm, right? Then at translational level, when it is getting synthesized as a protein, that level also we can control. Correct? So, regulation of gene expression, that refers to a very broad term and that may occur at various levels. So, considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. So, in eukaryotes, that regulation could be extracted at transcriptional level. Transcriptional level in the sense formation of the primary transcript. Formation of primary transcript. This level we can do. Then, second one, processing level. That means regulation of splicing. That we can do. Regulation of splicing. Then, third one, transport of mRNA from nucleus to cytoplasm. mRNA from nucleus to cytoplasm. Okay. Then, fourth one, translational level. Translational level also we can control the gene expression or we can regulate the gene expression. Transcriptional level, processing level, transport of mRNA from nucleus to cytoplasm, then translational level. In all these levels, we can control the gene expression or we can regulate the gene expression. Okay. Next, in a particular organism, in the cell, the genes will be having synthesized proteins. And these expressed proteins that may have set of functions also. Got it? In a particular organism, in that cell, in the cell of an organism, the genes will be, those genes will be synthesized with particular protein. And these expressed proteins, these synthesized proteins, they'll be having a single function or a set of functions. Okay, both chances are there. There may be a single function or maybe a set of functions. In an organism, in the cell, protein synthesizing will take place and these synthesized proteins will be having a single function or a set of functions. So, for example, we are talking about, here we are going to explain E. coli. E. coli, you know, in E. coli, one enzyme, that enzyme is called as beta-galactosidase. Beta-galactosidase enzyme. It is synthesized with E. coli. Note down, this is very, very important for MCQ and for board exam also. This lab operon concept. This is the basic concept which I am going to explain now. So, if you are understanding this E. coli, this one, it will be easy for you. Okay, that's how the regulation of gene expression is taking place. From this example, it will become very clear. Okay, so for example, in E. coli is an enzyme. There is an enzyme called beta-galactosidase. Note down the enzyme beta-galactosidase, which is synthesized in E. coli. So what is the function of this beta-galactosidase enzyme? The function of this beta-galactosidase enzyme. This beta-galactosidase enzyme catalyzes the hydrolysis of lactose and it forms glucose and galactose. Okay, so beta-galactosidase enzyme, this lactose hydrolysis, okay, it is taking place in E. coli in the presence of which enzyme? Beta-galactosidase enzyme, and it is forming glucose and galactose. Lactose is getting the hydrolysis of lactose is taking place and it forms glucose and galactose in the presence of an enzyme. Which enzyme? Beta-galactosidase enzyme. So this lactose, which is a disaccharide, lactose is a disaccharide, it is getting hydrolyzed into glucose and galactose with the help of beta-galactosidase enzyme. Lactose is a disaccharide and it is getting hydrolyzed into glucose and galactose with the help of beta-galactosidase enzyme, which is present in E. coli. Okay, so this E. coli bacteria that uses glucose and galactose as a source of energy. Okay, so if there is no lactose around this bacteria, because it is hydrolyzing lactose and it is forming glucose and galactose in the presence of beta-galactosidase enzyme. So E. coli bacteria uses glucose and galactose as a source of energy. So if there is no lactose or presence of lactose is not there around this bacteria, means there is no need of conversion of this into glucose and galactose. So there is no need of the production of beta-galactosidase enzyme also, right? So from this example, what we can learn? The expression of a gene that helps the formation of that particular product, right? If a gene is getting expressed, that helps in the formation of that particular enzyme, right? So that metabolic or environmental or physiological conditions, these conditions, metabolic condition or environmental condition or physiological condition, all these conditions, if any of these conditions are present, that regulates the gene expression or that creates the situation for that particular gene expression, right? If lactose is not present, there is no need of hydrolysis. So hydrolysis, if they don't want to do the hydrolysis, no need to synthesize the beta-galactosidase enzyme, right? So there is no formation of that enzyme. So there is no need of expression of any gene which is coding for that beta-galactosidase enzyme synthesis, right? So that's all. All these are connected. So metabolic or environmental or physiological condition, these are regulating the expression of a gene, or that is controlling the expression of a gene or a gene expression. So how an embryo, an embryo, after the formation of an embryo, how it is getting differentiated and how it is developing into a complete organism. All these are related, right? All it is based on the regulation, coordinated regulation and expression of several genes. It's not with the help of only one gene, it is with the help of several genes. It is working in a coordinated manner and several set of genes are involved. They are coordinating and it is in a coordinated manner they are working, and an embryo is getting differentiated and it is forming a complete organism, right? So the genes in a cell, they are expressed to perform a particular function or a set of functions. For example, an enzyme called beta-galactosidase is synthesized by E. coli. So it is used to catalyze the hydrolysis of a disaccharide lactose, and it is getting converted into glucose and galactose. So the bacteria use them as a source of energy. This bacteria E. coli is using this glucose and galactose as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta-galactosidase. Okay. So, as an energy source, the bacteria do not have lactose around them. So, around that bacteria, there is no energy source or there is no lactose as an energy source. They won't use, or there is no need of this enzyme synthesis. Which enzyme? Beta-galactosidase enzyme. Got it? So, therefore, it is a metabolic, physiological or environmental conditions that regulate the expression of genes. Okay. So the development and the differentiation of embryo into adult organism are also a result of the coordinated regulation of expression of several set of genes. So note down all these points as such. I have made notes very short because that was only required for your exams. So it will be easy for you people also. So hope it's clear. Next, in prokaryotes. In eukaryotes, we have already said we can regulate gene expression at several stages. But in the case of prokaryotes, the main or the predominant site for the control of gene expression, that is by controlling the rate of transcriptional initiation. Okay. So in prokaryotes, how we can control? Transcriptional initiation we can control, and thereby we can regulate the expression of a gene. So how it is done in prokaryotes? Do you have any idea? You know that RNA polymerase helps in transcription, right? Here we said transcriptional initiation we can control, and that's how we can control regulation of, or that's how we can regulate the expression of a gene in prokaryotes. So RNA polymerase, that is helping in transcription, correct? So during transcription, RNA polymerase binds at a given promoter region, and that we have already learned about transcription, that RNA polymerase enzyme will come and bind in the promoter region, and that starts the, or that promotes the transcription process, right? So this should be regulated. In prokaryotes, this step only we can regulate. So this should be regulated to control the gene expression. So how it is getting regulated in prokaryotes? By the interaction with other proteins in this site. How it is getting controlled? By the interaction with other proteins in that site. Okay. So those type of interactions by accessory proteins in that enzyme binding site. Okay. See, we have already told RNA polymerase, it will come and bind at given at a particular promoter region or a given promoter region, and that starts the transcription process, right? So we can control how that protein expression we can control only by regulating this initiation of this transcription process. So what we can do, how we can control? By interaction with other protein which are present in this site, in this promoter site. Okay, so those type of interaction, how that interaction by other proteins or accessory proteins in that particular site, in that enzyme binding site, that affects the ability to recognize that enzyme binding site. See, if other proteins are coming and creating a problem there, or creating that other proteins are interacting with that promoter site, this RNA polymerase will find it difficult to identify that promoter region, or that cannot interact with that promoter site or that enzyme binding site, right? So that's how the regulation is done. So these type of proteins, they can act in both ways. Some other proteins, the accessory proteins which are present in that site, those accessory proteins are coming and interacting with this promoter or that primer, which site? That enzyme binding site. RNA polymerase will find it difficult to identify that enzyme binding site because other proteins will be there, right? So in such situation, that kind of proteins, they can act in both ways. Either they can act as activators, or they can activators in the sense they can act in a positive way. They can act as activators, or they can act as repressor. Repression means that is a negative way. Activator means in a positive way, this kind of accessory proteins can work. Or sometimes it can work as repressor. That means negative way. So in prokaryotes, DNA, in prokaryotic DNA, the accessibility of promoter region, it is regulated by the interaction of that protein with some sequence. Okay, those sequence, they are called as operators. Note down, in prokaryotic DNA, the accessibility of that promoter region, how that promoter region, is it accessible or not, that is regulated or that is controlled by interaction of protein with some sequence. This sequence is all called as operators. Okay, and the operator region is very near to the promoter element, and in most of the cases it will be very near. Mostly that operator region will be near to the promoter element, and in some other cases the operator sequence may bind with the repressor protein. Okay, it may bind with the repressor protein. That means repressor means that shows negative effect. Got it? So with an example, I will explain the next steps. So, and the important point is that each operon has specific operator and specific repressor. Each operon that has its specific operator and specific repressor. Okay. For example, lac operator. Note down, lac operator. That is present in the lac operon. Okay. Lac operator, it's present in the lac operon. Like that, it interacts specifically with lac repressor. Okay. Lac operator and lac repressor only. Okay, here you can see in prokaryotes, control of the rate of transcription initiation is the predominant site for control of the gene expression. How we can control? We can control the gene expression in prokaryotes by controlling the rate of transcriptional initiation. Okay, so in a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affects its ability to recognize start sites. Got it? In a transcriptional unit, RNA polymerase that will get its activity at a promoter region. So This we can regulate. How we can regulate? By interaction with accessory proteins. That can interact with the accessory proteins, and that interaction which affects the ability to recognize the start site. That interaction, that accessory protein interaction will affect the ability to recognize the starting sites. Okay. So these regulatory proteins, this can act both positively and negatively. This regulatory proteins, they can act in positive way. They can work as activators and they can work as repressors. Repressor means they can work as activators in a positive way, or it can work as repressor. Repressor in the sense they can work in a negative way. So the accessibility of promoter region of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed operators. Okay. So the accessibility of promoter regions of the prokaryotic DNA, it is regulated by interaction of proteins with sequences. That sequences which are called as operators. So that operator region is adjacent to the promoter element in most operons. In most of the operons, that operator region will be adjacent to the promoter element. And in many cases, the sequence of the operator binds a repressor protein. That sequence of that operator that binds a repressor protein. Okay. So each operon has its specific operator and a specific repressor. So each operon, that is very important. Each operon, it is having specific operator and specific repressor. So for example, LAC operator. LAC operator is present only in the LAC operon and that interacts specifically with LAC repressor. Got it? LAC operator. LAC operator will be present in the LAC operon and that LAC repressor only will work in this LAC operon. Got it? Next is in LAC operon. LAC operon. LAC means this is the short form of lactose. Okay. In LAC operon, a polycistronic structural gene. Structural gene we have already learned. It is the expressing gene. That structural gene is regulated by a common promoter and regulatory genes. So in LAC operon, a polycistronic structural gene is regulated by a common promoter and regulatory gene. Here you can see the promoter and this is the operator. These are polycistronic structural genes. These are the expressing genes in the LAC operon. Okay, here this is the operator and this is the promoter region. Okay. So such arrangement, it is very common. The polycistronic structural gene is regulated by a common promoter and regulatory genes. So these type of arrangements, they are common in bacteria. This type of arrangements are common in bacteria. It is called as operon. This kind of arrangements are common in bacteria and it is called as operon. For example, LAC operon, it is an example for operon, that concept, operon concept, and trip operon, then val operon, then his operon. All these are examples for operon concept. So from in that, we are going to learn about LAC operon in detail. Okay. Here, this is the LAC operon concept, lactose. Okay. LAC means lactose. Don't get confused. LAC operon means lactose operon concept. So LAC operon concept. Here you can see this is the structure Z, Y, A. These are the structural genes, or these are the expressing genes. So in absence of inducer, here repressor, this repressor binds to the operator region. Here you can see the repressor first. This inducer, it forms the repressor mRNA and forms the repressor, and repressor binds to the operator region. Here this is O, this is the operator region, and that prevents RNA polymerase from transcribing the operon. It prevents RNA polymerase. Okay, and that prevents the transcription. Okay, but here in the presence of inducer, you can see repressor mRNA is forming, and from that inactive repressor, here inducer is there. In the presence of inducer, that repressor is getting, or it is becoming inactive. Then transcription, it is taking place, it is forming beta galactosidase, then permease, and here translation is taking place. This protein formation is taking place. That's what I mean. So we are going to discuss in this about this lac operon in detail. So here the lac operon concept that we are going to learn in detail. So I'll explain this lac operon concept in detail. So lac operon means it is having one regulator gene and three structural genes. See, one regulator gene and three structural genes. Which are the structural genes? Z, Y, and A. When you are getting a question to write about the lac operon concept for your board exam, you have to write, mainly you have to write these points with this diagram. Okay, you have to write, you have to draw this diagram, and you have to write these points mainly. Okay, then all these points are very important for your NEET MCQs. So you can't avoid this, or you can't escape, or you can't skip this topic. Okay, so the lac operon concept, it is having only one regulator gene and three structural genes. These are the three structural genes, Z, Y, and A. Three structural genes. Okay, so here you can see the I gene. This I gene that codes for repressor of lac operon. Repressor of lac operon. This I gene codes for repressor of lac operon. Okay. This is the repressor. Repressor mRNA is forming, and this is the repressor. Okay. And Z, here you can see this is the Z gene. This Z gene codes for beta galactosidase. Z gene codes for beta galactosidase. Note down each gene with the enzyme. Beta galactosidase. It is coded by Z gene, and it is responsible. This Z gene is responsible for the hydrolysis of lactose into glucose and galactose. You should remember the role of each gene. Z gene is coding for beta galactosidase, and this beta galactosidase is responsible for the hydrolysis of lactose into glucose and galactose. Got it? Beta galactosidase is an enzyme, and it is hydrolyzing a disaccharide lactose. Lactose is a disaccharide, and it is getting hydrolyzed into glucose and galactose in the presence of which enzyme? Enzyme beta galactosidase. It is coded by Z gene. Okay. Next, Z gene over. Next is Y gene. Y gene codes for permease. Permease. Okay. Y gene codes for permease. And what is the role of permease? This permease increases permeability of the cell to beta galactosidase. Okay. What is the role of permease? Permease increases the permeability. From the name itself, it's clear, right? Permeability of the cell to beta galactosidase. This beta galactosidase should enter inside the cell. Correct? Then only it can help for the hydrolysis of lactose. So the beta galactosidase should enter inside cell. So that the cell should permit the entry of beta galactosidase. So this Y gene forms permease, which increases the permeability of the cell to which enzyme? That increases the permeability of the cell to the beta galactosidase enzyme. So beta galactosidase will be permeable inside the cell only after the production, or that increase, this permease enzyme increases the permeability of the cell. For which enzyme? For beta galactosidase enzyme. And next is A gene. Okay, A gene. This A gene that encodes trans-acetylase. Trans-acetylase. A gene encodes trans-acetylase. Here you can see trans-acetylase. What is the role of this? These are the enzymes transferring an acetyl group from acetyl coenzyme A to galactosidase, lactosidase and glucosidase. Okay, this is enzyme transferring an acetyl group. This is the enzyme which is transferring acetyl group from acetyl coenzyme A to galactosidase, lactosidase and glucosidase. Okay, so all these three products, they are required for the metabolism of lactose. Got it? So Y gene codes for permease that increases the permeability of cell to beta galactosidase. Then next is A gene. A gene encodes a trans-acetylase. These are enzymes transferring an acetyl group from acetyl coenzyme A to galactosidase, lactosidase and glucoside. Sorry, lactoside and glucoside. So they are coded for by the A gene of lac operon in E. coli. Okay, note down, these are important for your NEET exam. Galactoside, lactoside and glucoside. Acetyl coenzyme A to galactoside, lactoside and glucoside. Note down all these three. So hence all three gene products in lac operon are required for metabolism of lactose, because each gene is having specific role. So in most of the operon as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Like this in all operon. We said few more examples for this operon, right? So not only in this lac operon, in other operon also they need to work together in the metabolic pathway. Got it? So lactose. This lactose is the substrate for the enzyme beta galactosidase, and that regulates switching on and switching off the operon. Right? If the lactose substrate is present, then that enzyme beta galactosidase will form, and that regulates the on and off of this operon. Right? If lactose is not present, means this operon won't work. If it is present, means it will get on. So it is termed as inducer. It is termed as inducer. Here, this I gene, inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of bacteria, the lactose is transported into the cell through the action of permease. Got it? What is the role of permease? See, in a carbon source, like glucose is needed for the growth of an organism. If glucose is not present, instead of that, lactose is provided. Okay, so what should happen there? That lactose should get hydrolyzed and it should form glucose and galactose. So for that, which enzyme is helping? beta galactosidase enzyme is helping. So there, this operon should work. Lac operon should work. Okay. So that lactose, how that lactose should get transported? It will get transported inside the cell with the help of permease enzyme because it increases the permeability, right? So that is transported into the cell through the action of permease. How it is working? First, beta galactosidase enzyme will form that will hydrolyze glucose and galactose. So this beta galactosidase enzyme permeability is increased by the enzyme permease. Then only it can work the hydrolysis. It can work for the process of hydrolysis. Okay. Then this lactose induces the operon in the following manner. I will explain how it is inducing the operon. The repressor of the operon is synthesized from the I gene. Here you can see the I gene, the repressor of this operon. That is synthesized from the I gene. And the repressor protein, here the repressor protein binds to the operator region. That repressor protein, here you can see the repressor mRNA. I gene induces the synthesis of repressor. And repressor protein is binding to the operator region. Okay, repressor protein is binding to the operator region of the operon, and usually where the RNA polymerase is binding to the operator region, right? So that region, operator region, this repressor protein is getting bound to that operator region, and RNA polymerase won't find a place to bind, so the RNA polymerase prevents the, so that bound repressor protein that prevents the RNA polymerase from transcribing the operon. So these genes transcription won't take place because which region? The operator region. In the operator region, you can see a repressor protein. So that repressor protein will block the binding of RNA polymerase. So that time RNA polymerase won't be able to find out a binding region because there this repressor protein is there. So that blocks the transcription or transcribing process of the operon. So in the presence of an inducer such as lactose or allolactose, that repressor is inactivated by interaction with the inducer. Okay, so in the presence of an inducer, lactose or allolactose, the repressor is inactivated by the interaction with the inducer. Okay, so this allows RNA polymerase access to the promoter and transcription proceeds. Here the repressor mRNA, it is forming a repressor. But when here inducer is absent, but here in the second case inducer is present. So this inducer makes the repressor in an inactive form. So that repressor can't work there. So that time this RNA polymerase will find the space for binding, or it will bind to the binding region, so that the transcription process will work smoothly. And this mRNAs will form and it will synthesize the particular proteins, or the translation also will take place. Okay, so the regulation of lac operon, that can also be visualized as a regulation of enzyme synthesis by the substrate. Okay, so these genes are synthesizing the enzyme by the substrate. So that also we can see here in the case of lac operon concept. Okay. So remember, glucose or galactose cannot act as an inducer for lac operon. Okay, glucose or galactose cannot act as an inducer for lac operon. So regulation of lac operon by repressor, it is referred to as negative regulation because it is blocking that effect, right? So it is a negative regulation. Lac operon is a negative regulation of gene expression. And lac operon is under control of positive regulation as well. Okay, it is under control of positive regulation also, but mainly it is referred as a negative gene regulation. Okay. Hope operon concept is clear. So next is human genome project and fingerprinting, DNA fingerprinting and all that we can see in the next class. Hope everything is clear up to this. Do you have any doubt? Is it clear? You want me to repeat anything? Oh, thank you. We can see in the next class.
Thank you, ma'am.